Hello friends, today we will learn about growth, nutrition and metabolism of bacteria. The minimum nutritional requirement for growth and multiplication of bacteria includes sources of carbon, nitrogen, hydrogen, oxygen and some organic salts. In this video, we will discuss about bacterial cell division, generation time, bacterial count, bacterial growth curve, continuous culture, bacterial growth in vivo, bacterial nutrition, bacterial metabolism. All these points we will cover in this video. So let's begin. Bacteria can be classified on the basis of nutrition. Let's know about this nutrition classification. So first is phototrophs. So phototrophs. Phototrophs means these derive energy from the sunlight. Chemotrophs. Second is chemotrophs. That means these use chemicals for deriving energy. Third is autotrophs. Autotrophs means bacteria which can synthesize their essential metabolites from the atmospheric carbon dioxide and nitrogen. Fourth is heterotrophs. These depend on the preformed organic compounds. Majority of the pathogenic bacteria are heterotrophs and parasitic. They have evolved with the animal body where ready-made nutrients are freely available. And now we will discuss about bacterial growth. In this, we will first discuss about bacterial cell division. Bacteria can be divided by binary fission. When a bacterial cell reaches a critical mass in its cellular constituents, the cell division starts. Bacterial nucleus or chromosome. Bacterial nucleus or chromosome is a circular double-stranded DNA molecule. Nuclear division precedes bacterial cell division. During replication, the two strands of DNA are separated and now complementary strands are formed. Thus, two identical double-stranded DNA molecules are formed. The replicated DNA molecules are distributed to the two daughter cells. A transverse septum grows across the cell from the cell membrane. This is followed by the deposition of cell wall materials and the two daughter cells separate. In some bacterial species, the daughter cells may remain partially attached even after cell division. Now let's know about generation time. The time required for a bacterium to give rise to two daughter cells under optimum conditions is known as generation time. The generation time in most of the medically important bacteria is about 20 minutes. In Mycobacterium tuberculosis it is about 20 hours and in Lepra bacilli it is 20 days. As bacteria grow so rapidly and by geometric progression, a single bacterium can be theoretically give rise to 10 to the power 21 progeny in 24 hours. In actual practice, bacterial multiplication is arrested after a few cell divisions due to exhaustion of nutrients and accumulation of toxic products. Now let's know about bacterial count. First we will discuss about total count. What do we mean by total count? This indicates total number of bacteria in the specimen and irrespective of whether they are living or dead. Total count may be made by counting the bacteria under microscope using counting chamber. Now in second point is viable count. What do you mean by viable count? Let's know about viable count. It measures the number of living cells. That means viable cells. Viable count are obtained by dilution or plating methods. In the dilution method, several tubes with liquid culture media are inoculated with varying dilutions of sample and the viable count calculated statistically from the number of tubes showing growth. Though this method does not give accurate values but is still used widely for finding out presumptive coliform count in drinking water. In the plating method, appropriate dilutions of the specimen are inoculated on solid media, either on the surface of plates or as pore plates. The number of colonies that grow after incubation gives an estimate of the viable count. Now let's know about bacterial growth curve. When a bacterium is inoculated into a suitable culture medium and incubated, its growth follows a definite course. When bacterial count of such culture is determined at different intervals and plotted in relation to time, a growth curve is obtained. The growth curve has four phases. Let's know about four phases. First phase is lag phase. After Now let's know about lag phase. After inoculation of the culture medium, multiplication usually does not begin immediately. The period between inoculation and beginning of multiplication is known as lag phase. During this period, the organisms adapt to the new environment, during which necessary enzymes and intermediate metabolites are built up in adequate quantities for multiplication to proceed. There is increase in the size of the cells, but there is no appreciable increase in numbers. The duration of lag phase varies with the species, nature of culture medium and temperature, etc. Now let's know about log phase. Log or exponential phase. The cell division starts and their numbers increase exponentially or by geometric progression with time. If the logarithm of the Babel count is plotted against time, a straight line is obtained. Third point is stationary phase. After log phase, the bacterial growth ceases almost completely due to exhaustion of nutrients and accumulation of toxic products. The number of progeny cells formed is just enough to replace the number of cells that die. The number of viable cells remains stationary as there is almost a balance between the dying cells and the new formed cells. Now let's know about phase of decline. It's the fourth phase, last phase. So after a period of stationary phase, the bacterial population decreases due to the death of cells. The decline phase starts due to exhaustion of nutrients, accumulation of toxic products, and autolytic enzymes. There is decline in viable count and not in total count. With autolytic bacteria, even the total count shows a phase of decline. Now let's know about morphological and physiological alterations of cells during growth curve. First is the end of the lag phase. That means bacteria have maximum cell size. In the log phase, what happens? In the log phase, cells are smaller and stain uniformly. In stationary phase, cells are variable and show irregular staining. Sporulation occurs. Many bacteria produce exotoxins. In the phase of decline, involution forms are common. Now let's know about continuous culture. When bacteria are grown in a vessel of liquid medium, after some time the cell division ceases and multiplication is arrested due to depletion of nutrients or accumulation of toxic products by the use of special devices for replenishing nutrients and removing bacterial cells, continuous culture of bacteria is maintained. The technique, namely chemostat and turbidostat, is in use. Maintenance of continuous culture of bacteria is sometimes required for industrial and research purpose. Now let's know about the bacterial growth in vivo. There is a significant difference of bacterial growth in human body and artificial culture medium. When bacteria multiply in host tissues, the situation may be intermediate between a batch culture and a continuous culture. The source of nutrients may be plenty, but the defense mechanisms of the body influence the bacterial growth in vivo. Now let's know about bacterial nutrition. The principal constituent of bacterial cell is water, which is about 80% of the total weight. For growth of bacteria, the minimum nutritional requirement of is water, a source of carbon, a source of nitrogen and some inorganic salts. These nutritional elements may be supplied in suitable artificial culture media. In addition to these, bacteria require sulfur, phosphorus, and other elements such as sodium, potassium, magnesium, iron, and manganese in small amounts. Some bacteria are fastidious and will not grow unless certain organic compounds are added to the medium. These are known as growth factor or bacterial vitamins. In many cases, bacterial vitamins are same as the vitamins necessary for mammalian nutrition, particularly those belonging to the vitamin B group. Thiamine, nicotinic acid, riboflavin, pyridoxine, folic acid, and vitamin B12. These are the all belonging to vitamins B group. Okay, so these growth factors may be essential for some bacteria when growth does not occur in their absence, or accessory when they enhance growth without being absolutely necessary for it. Now let's know about environmental factors affecting growth. First is moisture and desiccation. Moisture is an essential requirement for the growth of bacteria because 80% of the bacteria cell consists of water. However, the drying has varying effects in different organisms. Some organisms like Treponema pallidum and N. gonorrhea die quickly after drying, while M. tuberculosis and Staph aureus may survive for several weeks. And drying in cold and vacuum is used for preservation of microorganisms. Now let's know about the oxygen. On the basis of requirement of oxygen, bacteria are divided into aerobes and anaerobes. Let's know about aerobes. They require oxygen for their growth. They may be obligate aerobes, which grow only in the presence of oxygen, or facultative anaerobes that are ordinary aerobes but can also grow without oxygen. Most of the pathogenic bacteria are facultative anaerobes. Microaerophilic bacteria can grow in the presence of traces of oxygen. Campylobacteria bacter is an example of microaerophilic bacteria. Now let's know about anaerobes. The obligate or strict anaerobes can grow only in the presence of oxygen. Clostridium tetani is one such example. Now let's know about carbon dioxide. Third point is carbon dioxide. Third factor is environmental factor is carbon dioxide. Small amount of carbon dioxide is required by all bacteria. This requirement is usually made available by the carbon dioxide present in the atmosphere or produced endogenously as a product of cellular metabolism. Some organisms such as Brucella abro- abortus require much higher level of carbon dioxide, that means 5 to 10% of their growth, especially for primary isolation. Now let's know about fourth factor, that is temperature. The optimal temperature range varies with different bacterial species. Most of the pathogenic bacteria grow optimally at 37 degrees centigrade. Our body temperature, that means our body temperature, on the basis of optimum temperature range for growth, bacteria are grouped into various types. First is mesophiles, that is between 25 degrees centigrade to 40 degrees centigrade. Example, majority of the pathogenic bacteria. Psychrophils, that is below 20 degrees centigrade. Example, soil and water saprophytes. Thermophiles, that is between 55 degrees centigrade and 80 degrees centigrade. Example, Bacillus stereothermophilus. And now let's know about another factor, that is pH. Most of the medically important bacteria can grow at pH 7.2 to 7.6. However, some bacteria such as lactobacilli and cholera vibrio grow at acidic and alkaline pH respectively. Now let's know about another factor, that is light. Bacteria that is that can grow well in darkness. They are sensitive to ultraviolet rays and other radiations. Photochromogenic mycobacteria produce pigment only on exposure to light. Now let's know about another factor, osmotic effect. Bacteria are able to withstand a wide range of external osmotic variation because of the mechanical strength of the cell wall. Sudden exposure of bacteria to hypotonic solutions may cause plasmolysis, osmotic withdrawal of water leading to shrinkage of protoplasm. This occurs more readily in gram-negative than in gram-positive bacteria. Sudden transfer to distilled water may cause plasmoptosis, excessive osmotic imbibition of water leading to swelling and rupture of the cell. Now let's know about mechanical and sonic stresses. Though bacteria have tough cell walls, they may be ruptured and disintegrated by vigorous shaking with glass beads and by exposure to ultrasonic vibrations. Now let's know about bacterial metabolism. The series of changes of substances like carbohydrates, proteins and fats within the bacterial cell from ab- absorption to elimination is known as metabolism of the substances. Aerobic bacteria obtain their energy only through oxidation involving oxygen as the ultimate hydrogen acceptors, while the anaerobes use hydrogen acceptors other than oxygen. Now let's know about the oxidation. In case of aerobes, where the ultimate hydrogen acceptor is oxygen, the carbon and energy source may be completely oxidized to carbon dioxide and water. The conversion of adenosine diphosphate, that is ADP, to ATP, adenosine triphosphate, means adenosine diphosphate to triphosphate, provides energy in this process. This process is called oxidative phosphorylation. Small amount of hydrogen peroxide produced in the process is disposed of by enzymes, catalase or peroxidase present in aerobes. Now let's know about fermentation. In anaerobic respiration, hydrogen acceptor compounds are nitrates or sulfates instead of oxygen. In fermentation, a series of oxidoreductions occur in which the carbon and energy source acts as both the electron donor and electron acceptor. The organic end products of fermentation include acids, that is lactic acid, formic acid, pyruvic acid, gas, hydrogen, carbon dioxide and oxygen, and alcohols. During fermentation, energy rich phosphate bonds are produced by the introduction of organic phosphate into intermediate metabolites. This process is known as substrate level phosphorylation. These energy rich phosphate groups so formed help in conversion of ADP to ATP. Fermentation is carried out both obligate and facultative anaerobes. The oxygen and hydrogen peroxide are toxic to anaerobes. The reason for this toxicity is not well understood. It is suggested that in the presence of oxygen, hydrogen peroxide accumulates in the media and inhibits the growth of anaerobes. The enzyme catalase, which splices hydrogen peroxide, is present in aerobic bacteria but is absent in the anaerobes. Another reason might be that anaerobes possess essential enzymes that are active only in the reduced state. The facultative anaerobes may act in both the ways, that is, by the process of fermentation and aerobic respiration. They derive their energy by fermentation in the absence of oxygen but switch over to aerobic respiration in the presence of oxygen. Now, let's know about oxidation reduction potential, or in short, redox potential, or OR. The oxidizing or reducing condition of a system is indicated by the net readiness of all the components in that system to accept or to part with the electrons. This is called oxidation reduction potential or redox potential. The redox potential of a medium is determined by recording the electrical potential difference set up between the medium and an unattackable electrode immersed in it. The electrode potential, in short EH, can be measured in millivolts. This depends upon the state of oxidation or reduction of the solution. The more oxidized the system, the higher is the potential, and in reduced system potential is lower. The redox potential of the medium can also be assessed by the use of certain indicator dyes such as methylene blue or rizazurin, which become colored in oxidized state. It is simple but less accurate method. Anaerobic organisms require low redox potential for their growth. Finally finished. Any questions regarding this, you can ask me in the comment box, and subscribe my channel to know more updates. Thank you.